guess I was trained as an engineer, and I wanted to really understand the brain in a very intuitive way. Could you really see a thought or a feeling, but not lose sight of all of the building blocks, molecular, chemical, electrical, that made it up? And so I've spent the last, I guess, 2 decades roughly, running a technology inventing group at MIT, where we've been building tools to try to help reveal those building blocks without losing sight of the emergent whole. And so the tools we've been building include ways to control high-speed signals of the brain, recording high-speed signals of the brain, and mapping molecules, wiring, and connections. And we share all the tools freely, but I drew in some orange arrows here because increasingly we've been thinking about integrating all the tools into a cohesive workflow. Now, if you could control a brain down to every cell and record every cell in a brain and map all the wiring, that's going to be a huge data set. And so increasingly, we've been thinking that our goal needs to be to focus on making computer simulations of the brain. And many people, of course, are working on modeling of the brain, and there's a lot of amazing pioneers of that field here. One of the things that we're trying to figure out is what kinds of ground truth-oriented data might allow us to be able to simulate brain circuits in terms of their building blocks so that we can reveal what mechanisms contribute to emergent functions. So the tool sets we've been building over the years include our method for controlling brain cells down to the single cells across the brain. This is a way that we control the brain with light called optogenetics. And so what you're seeing here is a single cell green algae, and it's swimming around. And we're about to zoom into a little eye spot there, which is chock full of light-activated proteins. These are proteins that convert light into electrical signals so that that single-celled algae can swim around and get near the surface of a body of water and photosynthesize better. And what this eye spot has are proteins that, well, open up little pores or channels when exposed to light. So you can, when sunlight hits these proteins, it'll open up a little hole and you'll get positive charge flowing across the membrane of the eye spot. And that causes an electrical signal that through a cascade of molecular downstream impacts will eventually control those little tails, the flagellae that help the critter swim. And so what we've done is to take the DNA that encodes for this protein. There's a lot of serendipity in this story. It turns out it's a small chunk of DNA. You can pop it into a brain cell with off-the-shelf gene therapy methods. And now the brain cell will manufacture that protein and install it properly, and it'll function. And you can then shine light on that brain cell, and what will happen is that it'll fire off electrical potentials, not unlike the ones that you're hearing that are happening in your brain right now as you hear me say these words. So in the years since, we've been working on trying to develop tools that flesh out the 3 legs of that tripod. And one thing we've been working heavily on is voltage imaging. This is like the opposite of optogenetics, if you will. You could record electrical activity with a wire or other amazing tools that can pinpoint individual cells, but how do you do it at scale across an entire brain? And so we've been building what might be some of the world's fastest microscopes, taking advantage of advances in camera technology. So as long as cameras are driven by cell phones, maybe you didn't need to go faster than 100 frames a second, but in a self-driving car drone era, low light condition, 1,000 frame per second imaging needs to be affordable. And so we can image the voltage of cells across a larval zebrafish, which is with about 100,000 neurons, expressing a molecule that sort of operates like the opposite of what I just told you. And what you see here look like little electrical traces, but they're not. They're being picked up on a camera. And we have been able to increase the number of voltage image neurons by a factor of over 1,000 in just a couple years. And then finally, mapping the brain. Now, there are lots of ways of nanoimaging, but how do you scale them to an entire brain? And one of our favorite strategies in the group is to think about doing the opposite of what people are doing. And so we wondered, for 300 years, the way you image in biology is you magnify an image with a lens. Let's magnify the brain itself. And so the basic idea is to install baby diaper polymer, a swallowable material inside the brain. So it's not a living brain at this point. But in panel B is a piece of the brain. Panel C, we blew it up. And this is a little movie just to show you half an hour sped up to half a minute of how it looks. So we polymerized the brain earlier, and we added water, and I hope you can see it swelling. So the hope then is to be able to look at brain circuits in great detail. Here's an example of a piece of the mouse brain expressing fluorescent proteins, and I hope you can see that the fine wiring of the brain can be visualized here. These are sort of bar-coded neurons, if you will, expressing combinations of proteins. There's an organization, E11, who I think some of you might have seen the little booth out front, which is looking to scale up the bar coding and help map even larger circuitry. So the hope then is to integrate these tools and to get live data shown in the blue upper left, molecular data shown in the yellow lower left, and to bring it all together. Can we infer from dynamics what will happen next in the brain? Can we go from the dynamics and infer the physiology? And then can we close the loop by mapping the molecules? So it's both top-down and bottom-up modeling. And so this is a very infant stage project, but we're hoping to begin with small brains like fish and worms, but scale to mice and perhaps even humans. So very excited about the time to come.